guys! So today we're going to start a new lesson. So this is day 1 of lesson 7.3. Today we're going to talk a lot about cellular transport and basically how certain substances can pass through the plasma membrane, right? Alright, so our focus question for this lesson is what processes enable substances to move into or out of the cell? As you can see here, like what we said before, small uncharged molecules can just pass straight through the membrane, no problem, because they can mix with the oily membrane inside and they can go fit straight through the plasma through the phospholipids, right? Charged molecules are completely rejected because even though they're very small, they can't mix with this oily internal environment of the membrane. And large molecules, which are also called macromolecules, this would be like a big protein or some type of substance that's really large, can't get through, not just because it could be charged, but because it can't actually squeeze between all of these little phospholipids, right? So in this lesson, we're just going to be talking about how different substances can enter and exit the cell. All right, so here are some vocab words. I'm sure you might have heard of some of them, but probably not all of them. And you guys remember that homeostasis means regulating a constant internal environment, meaning that the cell is smart. It knows what it needs. It knows how much of a substance needs to come inside, and it knows how much of a substance needs to leave. And it needs to maintain a perfect balance of what is entering and what is exiting the cell. So, in cell transport, we have two different types. We have active transport and we have passive transport. So passive transport is a type of transport that does not require any energy to happen. It's basically just free. It happens naturally. It's not a forced process, so you don't have to give any energy to make these things happen. 3 examples of these are diffusion, osmosis, and facilitated diffusion, which obviously we're going to get into in the next couple of days. This is not all happening today. Active transport, on the other hand, is a type of cellular transport which requires ATP or energy, which means that this is a forced process. It usually happens when we're dealing with charged molecules or with large molecules or even just molecules moving in a direction that normally and naturally they would not have been moving in, right? So passive and active. So at the beginning, we're just going to be talking about passive for a little while, and then we'll get into active later on this week. All right, so first thing we need to talk about is just regular old diffusion. As you guys saw in that lab that we were looking at yesterday, there were 2 different types of dots, and some of them could move straight through the membrane. If you're able to move straight through the membrane with no problems whatsoever, no protein required, this is called diffusion. Okay? So particles in solids, liquids, and gases are in constant random motion. Particles are always, always, always in motion. You guys remember this from when you learned about chemistry a long time ago, and you just know that particles never truly stop. Even when they're in their solid state, they're just vibrating in place, but they haven't actually stopped moving. So diffusion is the net movement of particles from an area of high concentration to an area of low concentration. Additional energy is not required for diffusion because these particles are already moving in that direction. So if you look over here, particles like to move from areas where they are in a high concentration, which means an area where there is a lot of substances that are similar to them, to areas where they are in a low concentration. You've got to think of particles as like antisocial people at a party. They don't want to be crowded up. They don't want to have 4 of them all together here. They would rather move in this direction where they can be free and happy and far away from all the other particles that are like them. This is basically a very natural way of movement. Particles do not like to be close to other substances that are like them. They are always going to try to move to an area where there is a lower concentration of them so that they can be free and move far away from everybody, right? So the same thing happens when I drop a little bit of food coloring into water. It is all going to separate out and become perfectly evenly mixed. So if I look at this is an example of food coloring in water. It's not just going to stay a drop like this. As time goes by and as all the particles continue to move and bump into each other, they eventually start to spread out and spread out and spread out until all the water is evenly colored with the dye. That means that every single part of this water is going to have the same quantity of dye that is mixed into it. So, in this process, the dye went from an area where it was in a high concentration outwards towards an area that did not contain the dye, to where there was a low concentration of dye. And they kept separating and separating and separating until they reached equilibrium, meaning that there was no longer an area of low concentration for them to really move into. They have perfectly spread themselves out here, and now they're at equilibrium. Okay? So, when diffusion occurs over a long enough time, concentration will become uniform and the solution will reach dynamic equilibrium. Now, the word dynamic means in motion or constantly changing. So, basically, what this means is that even though they have reached equilibrium and all the dye is now evenly spread throughout the water, they have not stopped moving. They're still moving, but they're still at equilibrium. Okay? All right, so if I look over here, if I mix two dyes into water, blue and yellow, eventually they're going to mix up all together. They're not going to stay one on this side and one on this side, and they're going to form basically a green substance where they're all perfectly and evenly mixed. Okay, so diffusion causes the ink to move from areas where they're at a high concentration towards areas where the blue is at a low concentration and the yellow is at a low concentration. So they move away from substances that are like them and more towards areas where they can't find other substances like them until they are perfectly mixed. Okay? And remember, this is dynamic equilibrium because even though they are now spread out evenly throughout all the water, they are still in motion. They didn't just stop moving. They're like, okay, I like this area, I'm going to stop moving. They're continuing to move around. All right, so now things that will make diffusion happen faster is increasing the temperature. That'll make the particles move faster, or the concentration, and decreasing the mass of a substance or the pressure, which we're going to just show you in this lab. All right, so if I look over here, I have two different substances, right? And I want to get them to diffuse into each other. But before I start mixing them together, I just want you guys to look at them separately. So let's see what happens to the substances when I increase the temperature of the blue molecules and I decrease the temperature of the red molecules. Look at what happened to them. The blue ones started to move faster and the red ones started to move slower. Let's decrease the mass of the blue particles now and see what happens. They start to move even faster, right? So now you can see the different things have caused these particles to move in different ways. So now let's see what happens when we mix them together. And I want you guys to keep an eye on this because this is going to show you if they're at equilibrium. So right now you can see that there are 30 blue on this side and 0 red on this side. And over here you can see that there are 0 blue and there are 30 red dots on this side. So let's just increase the temperature of the red so that we're not sitting here forever, and let's remove the divider. This means we're going to allow them to mix now. Let's see how long it takes for us to reach equilibrium. Once these two lines get really close to each other, that means that they have perfectly mixed. And almost there. See, we're almost perfectly at equilibrium, but as you can see, even though now they are pretty much evenly spread throughout the entire container, they haven't stopped moving. But that doesn't mean that all the red ones are going to move back towards this area and all the blue ones are going to move back here. That's never going to happen. They are now evenly mixed because they have went from an area where they are at a high concentration towards areas where they're at a low concentration. And then we reached dynamic equilibrium. So we've basically reached a point where on average these two sides are going to have approximately the same number, but all of the substances within this area are still in motion. That's why it's called dynamic equilibrium. All right, so now guys, that's the end of the day 1 video. Today you're gonna have a quiz, and if you want, you can go and try out that lab, play around with the settings, discover new things, and let me know what you find out. And remember, there's a quiz on Shobi, so go ahead and do that. Good luck, guys.